Hi everyone, in this video we are going to talk about the DNA structure and replication, which is related to HL students, not for SL. So it's chapter or unit 7. It's a kind of small unit. It includes three different sections. The first part is talking about the DNA structure and replication. Most of these structure and replication have been, and it's almost a repetitive topic, so we will move very fast. It's not something that you don't know actually. The essential idea is that the structure of DNA is ideally suited to its function. In the background you will see a very beautiful structure of a DNA as a kind of 3D animation or a structure, and this beautiful structure was discovered by two famous scientists that we are going to talk about them after this. And this beautiful structure actually makes the possibility of the DNA to store the information, and what it is doing. The reason is the complexity and the shape and the way that it has been already made by the function of the nucleotides, phosphorus, and bases. These are the elements of the DNA that make this beautiful structure, and it makes it possible for a very unique function that he has. This is the outline of this unit, understanding, application, and skills. If I read very briefly, you will see that most of them we have already discussed and you have heard about the definitions. For example, nucleosomes help to supercoat DNA. You know that nucleosomes are structures that has role in supercoating in DNA. Or DNA structure suggests that a mechanism for DNA replication. DNA polymerases can only add nucleotides to the 3' end of a primer. This is what we have repeatedly talked about, how DNA polymerase work and how the direction of the adding nucleotides is in the direction of 5' to 3'. And this is what have been discussed before. DNA replication is continuous on the leading strand and discontinuous on the lagging strand. This is also we have discussed that in the lagging strand it makes Okazaki fragments, but in the leading strand it goes continuously. And the way that you want to remember is that this is leading means leader, and leader are just doing perfect as like continuous thing with one continuous strand, but lagging, it lacks something, which means that it makes some fragments, and then those fragments are ligated at the end in order to make a continuous strand later, not at the beginning. DNA replication is carried out by a complex system of enzyme. Well, you have seen the enzyme complexity. That's a kind of DNA polymerase, and then including the complex other materials together that makes it possible for the enzyme and the chemicals such as nucleotides in order to do this replication properly and efficiently. Some regions of DNA do not code for proteins but have other important functions. This is something that we discussed as in differentiation, or something that shows us if DNA it is not that all the DNA is able to make translation or to make protein. There are only some parts of the DNA which is possible to be translated, and there are some other regions that are not translated. But what are the functions of those who are not getting translated? In this part, you will see that there are important functions for that. Rosalind Franklin and Maurice Wilkin investigation. This is the story that which made to the discovery of the DNA structure, and mostly this discovery happened because of the X-ray diffraction works that was done by Rosalind Franklin and Maurice Wilkin laboratory. And then we are talking about the DNA profiling, which is a very important topic to discuss, and analysis of experiment done by Hershey and Chase, that they proved that the DNA is responsible for genetic information, not the protein, and the utilization of molecular visualization softwares that you see that how those types of software can be used. Okay. We start with the analysis of results of Hershey and Chase experiment. Hershey and Chase discovered and proved that the genetic information is coming from DNA, not coming from protein. With this small explanation, I will pause the video and wait for you to a little think, what does that mean? For example, what does that mean that the DNA is responsible for inheritance? Why not protein? Have you thought about that? Pause the video. Okay, did you think well? What was your result of thinking? Isn't it interesting that how can you say that DNA is the genetic material, not protein is genetic material? Do you understand what does that mean? So Hershey and Chase, actually with the type of experiment that they did, they proved this hypothesis and this idea. If I want to explain here and here, these two boxes, it makes you a little confused. So let's go to watch the a video from the Hershey and Chase experiment, and then I will explain. You will listen to this video, and that would be much more easier for you to understand. This is a SWF file. If you click on the link, then you need to have a cool file viewer or any extension in order to be able to open the file. So let's watch together.
Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity, Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing p32 labeled deoxyribonucleotides. This resulted in a phage population with p32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the p32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Okay, if you watched the video, maybe for the first time it's a little confusing, but you need to watch several times in order to catch up what is going on. Basically, the idea is that they use two types of radioisotopes in order to differentiate between DNA and protein codes. And basically, the experiment is conducted using phage. This is phage. Phage is a type of virus that attacks a bacteria. So they use two different types of radioisotope in order to differentiate once the DNA, and another time in order to differentiate between DNA and the protein. And then, according to the experiment, at the end, they noticed that this is the DNA that can go inside the bacteria, not the protein can go, because the DNA, once it goes inside, it produces the protein code from this DNA. But the protein coat, which is outside of the bacteria, cannot go inside because it is not a radioisotope. So basically, if you observe a couple of times, then you can figure out exactly from the beginning. And that was the idea, that they proved that this is the DNA which goes inside the bacteria and not the protein. Because of that radioisotopes, they figured out the one that has been inside the bacteria coming from where. So I go back and replay it again. This is the first experiment. In the first experiment, neither protein coat nor the DNA from the phage was used for a radioisotope, and both of them are without any radioisotope. So protein coat remained outside and DNA material went inside. So experiment was started by culture or by producing the phage in a medium containing S35 radioactive isotope in the amino acids. So because the amino acids included the radioisotopes, all the proteins that was made or were made by the phage were radioisotope. So protein radioisotope shows that only coat protein with the red color and the DNA without any radioisotope. The important part is here. The phage which already had a protein coat radioisotope was led to attack the bacteria. What did you expect from this experiment? You see that only DNA coming inside the bacteria, not the protein coat, because they saw that the radioisotope couldn't go inside the bacteria. So the protein coat remained outside and DNA went inside. So this was the first step to say that the phage will send the DNA inside the bacteria, not the protein. what do you expect when the DNA is going inside the bacteria? You expect that DNA can make the coat protein by itself. What kind of coat protein by itself will be made? The coat protein without any radioisotope. So that's what happened. DNA were transferred inside the bacteria, and the coat proteins were made without any radioisotope because there is no radioisotope amino acid inside the bacteria. So you can see that all the phages were produced, use the DNA as a genetic information in order to replicate the coat protein. The next time, the experiment for the next time was using the radioisotope phosphate, which is going inside the DNA, so that they let the phage grow in a container that included phosphate radioisotope, and the phosphate radioisotope only goes inside the DNA and not going into the amino acids. And this radioisotope phosphate showed inside the DNA. So you can see that the number of the phage viruses that had been already produced all included the DNA radioisotope. Okay, guess what happens when these phages, including the radioisotope DNA, will attack the bacteria? Again, only DNA went inside, and the radioisotope DNA went inside, and then this DNA radioisotope could produce phages from the information that he provided. But the thing is, radioisotope appeared in the bacteria, not in the coat protein. So once you separate these two by the centrifuge technique, the first time the bacteria didn't have radioisotope and the coat protein had. Second time, bacteria has but coat protein doesn't have. And in both cases, you can see this radioisotope separation in a centrifugation technique by observation using the radioisotope. So one is going to the upper part, which is lighter, and the other one, which is bacteria and heavier, going to the down part. So at the end, this experiment showed that this is the DNA which has the genetic information. Why? Because that's the DNA is causing the production of the next generation of the phages, not the protein. Protein coat remained outside of the bacteria, were just gone. But that DNA, which is coming inside, started to produce. So this means that that's the DNA is responsible for production, for making the next generation of the phages. So that's why we are saying that that's the DNA which is related to genetic information, not the protein. Now you can see this slide and read by yourself, and you can understand exactly what's going on. It is explaining the radioisotopes, the separation, and the usage of the two different types of radioisotope. One for making protein as radioisotope by the usage of sulfate 35, and the other one using phosphorus 32 for making the DNA radioisotope. And that's the way that it's make it possible for separation between DNA and protein by observation of radioisotopes. And how do they separate? By centrifuge. By centrifuge, what happens during the centrifuge? Well, the one which is lighter and smaller goes to the top, which is called supernatant, to the top of the centrifuge, and that which is a little heavier, which is called pellet, is going down of the centrifuge, which is bacteria. So whenever you do the centrifuge, you will see the virus in the top and bacteria in the bottom. But the first experiment, the radioisotope is for the DNA, and the first experiment was the radioisotope was for protein coat, so it goes down. And in the second experiment, the radioisotope was for DNA, which again goes with the bacteria, and it goes down. So depending on the radioisotope goes with bacteria or which goes with the virus, then it shows that which one is going up and which one is going down the centrifuge tube. And that's the way that we understand this separation, and we can interpret the result of the experiment.